All right, this is a recording. It is September 24th. This is Biology 105. It is the year 2025, and here we are. All right. Thank you all for making it to class today. You just had your first exam yesterday. You are either feeling good that you've got this class under control, or you are feeling okay, or you are feeling poorly about your performance. And whatever situation you are in, there is a recommended course of action, all right? If you're feeling middling about your performance, you can come talk to me about maybe changing up your study habits, what you're focusing on. Realistically, just seeing that first exam, you now know what my exams look like, you know what difficulty level they're set at, and you'll probably see an increased performance as we move throughout the semester. All right? We had a few people who got hundreds. Great job, those of you who got 100%. I couldn't be prouder. All right? And yeah, here's our histogram of what the here's a histogram of what the scores sort of looked like. You'll see that we didn't actually fall under normal distribution. In fact, it was sort of skewed towards higher scores here. Okay? So if you're somewhere in this region, you want to come meet with me. Okay? If you got less than 70%, so if you're anybody who makes up these bars right here, I've got that extra credit opportunity for you where you can make back half the points that you miss, okay? You have to come and talk to me or organize a meeting with me. It takes about 5 minutes for me to give you the materials and explain the full assignment, but essentially it's this, okay? Ready? I give you your exam. I give you your Scantron. You can see which questions you missed. For each question you missed, you write me 3 to 5 sentences where you explain what you answered and why it was incorrect, and what you think the correct answer is and why. That's it. It's meant to make sure that you understand those concepts that you didn't understand on the exam, okay? That's it. I'll explain it to you, give you, like 2 weeks to do it. Most people tell me it takes them 2 hours at the most to complete that assignment. You get back half the points you missed. If you got a 30%, your score goes up to a 65%, and all of a sudden your grade in the course bumps up significantly. Yes, please. So here's the thing. You can do this. Thank you so much. Actually, great question, first of all. Thank you for asking that. Once you get the assignment, I want it back within 2 weeks. All right? You can do this once per semester, okay? So if you got like a 68% and you want to wait to see how you did on exam 2, you totally can. If you want to wait to do this assignment until, seeing if maybe you failed exam 2, then totally feel free to wait. However, if you're on this end of the histogram, like do it for exam 1. Get it done, do it here, get your score back up so that you can, be more, stable in the class. But if you're right here around that cutoff of the 70%, then maybe you want to wait and see if you do worse on a future one. Does that sort of make sense? How'd I do? Did that explanation make sense to people? Cool. So it's an extra credit assignment that you can do once per semester for any lecture exam that you got 70% or lower on. All right? Now remember, your score's out of 100. I'm sorry, out of 75, not out of 100. So, if you look at your grade book, where's my calculator? 75%, oh my gosh, is 56, okay? So if you got 56 points or fewer on the exam, you qualify to do the extra credit assignment. Question?
So is it 70 or 75?
Oh, sorry, it's 70%. Thank you, I'm so sorry. I got too caught up in, the number 75 was already in my head. My bad. You need 52. You get a 52 or lower. Because I'll tell you what, otherwise, if you got like an 80% on this and you did the extra credit assignment, which I wouldn't let you do anyways, you only go up by 10% on a single exam. It doesn't make a huge difference in your final grade. So this assignment is meant to help out people who, need to refresh on some concepts. Any questions about that? If you got under 70%, just come to my office and I'll re-explain everything. I'll get you all set with that assignment. I really would appreciate it if you could try to do it during office hours, okay? because there's going to be a lot of students coming and trying to set up, individual meeting times and all that. My schedule is very booked. I am teaching 5 different classes. It's too much, and it's going to be very difficult for us to organize times outside of office hours. But it'll take me 5 minutes to give you those materials, okay? Office hours are on the syllabus. I posted them before. Any questions at all? Okay, awesome. Let's go ahead and get moving back into the materials of cell structure and function. So I told you we'd dial back a little bit and hit these, hit these concepts a little better. We're not going to watch the videos that are embedded in this PowerPoint because I don't want to sit and watch them. And I don't feel like it doesn't feel like a college course if I'm having you watch a recorded video. I'll ask that if my explanation is a little lacking for you, you can find tour of an animal cell video, tour of a plant cell video, both embedded in the PowerPoint. Just download the file, open it in PowerPoint, watch these videos. They're fine. There's also tons of other sources out there for video tours of plants and animal cells. Okay. I need to get this alpha here. Oh my gosh. Oh no. Now it's subtitling me. Okay. There we go. So, my clicker, I left the receiver in the other classroom. I told you we'd talk a little bit about the cell as a factory. Let's orient. I'm grounding myself. After all that exam talk, we are back into course material. Right now, we are talking about eukaryotic cells and what they've got, okay? The prokaryotic cells are pretty simple, right? It's a little bubble. It's got some genetic material in there. It's got some ribosomes in there. It can make a bunch of proteins. That's what it does. Now, in the eukaryotic cell, there's all this compartmentalization based on all of these membrane-bound organelles. Together, these are called the endomembrane system. So it's a system of membranes inside of the cell. It includes the endoplasmic reticulum, the mitochondria, the Golgi apparatus, peroxisomes, lysosomes, all of these bubbles made up of phospholipid bilayers that are sort of interconnected by vesicles fusing and all these sorts of things, okay? It's all about compartmentalization so you can specialize and be more efficient. Instead of having a factory where it's like all of the components get dumped into the middle of the room and everybody scrambles and tries to build a car as fast as possible, you have sections that assemble specific pieces of the car, and then they all get put together. So here is the cell as a factory analogy, okay? I just like this image. It makes me feel nostalgic, so I continue to use it. So check this out. We've got the nucleus of the cell, which sort of serves as the headquarters. The nucleus protects the blueprints for whatever the factory's making, okay? So the nucleus is the headquarters. The DNA and chromosomes are the blueprints. They're all held within that separate chamber, which is isolated by the nuclear envelope, the headquarters, right there. Nice and isolated from all the workers, okay? Next, we've got mitochondria. Mitochondria are the powerhouses of the cell, right? Those things are going to process high energy molecules and generate ATP, all right? So the mitochondria sort of power the other things going on in the cells. Here it's simplified, showing what would you call that? Like a couple gears connected by a, I'm not an engineer, all right? Power's here, going there, all right? But the power that's generated from the mitochondria is actually used to drive all sorts of processes throughout the cell, not exclusively protein synthesis. These little boxes say protein on them. It's an old image, okay? So this number 4 is it's the factory where the proteins are being made. So it's really the ribosomes, but it's also the endoplasmic reticulum Because many of the ribosomes are embedded in the endoplasmic reticulum. Question. Why? Because it helps to, check this out, the endoplasmic reticulum is contiguous with the nuclear envelope, and so the mRNA that gets generated here can be shipped directly into the endoplasmic reticulum, so it gets more focused and directed. It's like having a hallway connecting two, so you've got two different departments in the factory that are closely interrelated, and so you have a hallway that connects them so that when you're walking with those instructions, you don't get distracted by the coffee machine. You know, it's direct connection into the endoplasmic reticulum. And then this also means, we'll get into it in a second, but the endoplasmic reticulum also helps to package those proteins in little boxes like this so that they can be shipped and organized better in the Golgi apparatus. Yeah, awesome. So we've got the ER, the ribosomes, that's where you make the proteins. They are the production site. Next, you've got food production in the kitchen, okay? Really only present in autotrophs, all right? So this is a site where you're making sugars, okay? And here, in my example, in autotrophs, you've got a chloroplast that is making sugars that are then being burned by the mitochondria to drive the processes within the cell, all right? Next, we've got the shipping department, and that's sort of, or the sorting department, and that's the Golgi apparatus, all right? So check it out. Blueprints are read, and using energy from the mitochondria, proteins are made and packaged in the endoplasmic reticulum. Then those packages of proteins get taken to the Golgi apparatus where they are sorted, maybe put into a new box, maybe the final finishing touches are put on them, okay? And now they can be directed to where they need to go throughout the cell, or maybe they're destined to leave the cell. So the thing that transports them are vesicles. The vesicles are like the little carts, the trucks that are driving around and carrying things. Remember, a vesicle is just a bubble of phospholipid bilayer with some stuff in it. Well, the vesicle doesn't have to, yeah, with some stuff in it. Let's not get too convoluted with it, okay? And those can fuse between components of the endomembrane system because everything's phospholipid bilayer. Okay, so yeah, great. I know I emphasized this already, just one second. I know I emphasized this already, but I really want to make it clear that plants have chloroplasts and mitochondria, right? They just make the sugars and then burn the sugars, as opposed to a heterotroph, which eats the sugars to get them and then burns them. All right? Yes, please. Yeah.
Because it doesn't really show right in the slideshow.
Totally. It just shows this, right?
It shows them like overlapped.
Yeah. If you want to take a picture of this, go for it, folks. Also, when you're looking at these these PowerPoints, strongly encourage you to open them in the PowerPoint software. All right, it just works better. Yeah. Question.
So, I might not just be catching on, but the food production / what would it be in organisms that are not autotrophs?
You don't have one.
Okay.
Yeah.
That's what I thought.
Yeah. It's for you. It's an actual kitchen where you cook up food.
Okay.
Yeah.
So, like, for plants, system chloroplast.
Exactly. That's exactly right. So yeah, this part isn't in all organisms.
Great. Thank you.
Of course. Yes, please.
I know we're going to get into this later, but for the process of photosynthesis that the chloroplasts carry out, it's always mentioned like it's glucose, that's the sugar, but are there different formulas for slightly different sugars like sucrose?
100%. And in fact, I'm going to go a little bit more in depth into photosynthesis than other intro bio classes do, because I'm technically a plant biologist, I'm a plant physiologist technically. And so really, what the process of photosynthesis does is it produces something called G3P as a molecule, which then goes into all of these complicated branching pathways and makes all sorts of things besides just sugars. So we usually say, oh, photosynthesis takes carbon dioxide and makes glucose. But it really makes G3P, which then goes through a metabolic reaction, and some of it is used to make glucose. All right, yeah, solid? Cool, and yeah, we will get into that. I'll try to keep it, I'll still make it very clear, like, what you're gonna be tested on and what is just so that you actually know what's going on. Yes? Oh, sorry. Well, I just wanted to say yes to you. Yes, to everything that you're doing right now, I support it. All right. So here's a video of a tour of an animal cell. There's a video of a tour of a plant cell. I already gave you the spiel about this. I used to play those. I just can't do it again. I can't do it again. All right. So here's a plant cell. And we talked about this briefly. The plant cell has a cell wall. That's a major structural difference. You're doing an osmosis lab this week, tomorrow. So you're going to talk about the cell wall because it leads to differences in how plant versus animal cells respond to being placed into solutions of different tonicity. Don't worry, you'll get all the details you need in your lab this week, which is all about osmosis. Awesome. Cool. We talked about this. Oh, there's also these pores called plasmodesmata. So one of the major, question, yes. Oh, thank you. I'm so sorry, yeah. This cell is a factory is for a general eukaryotic cell. This here is for a generalized animal cell. And you'll see that it has no cell wall. That's the big thing. And it's got no central vacuole, and it's got no chloroplasts in it. If we skip over to the plant cell, here we've got a cell wall, we've got a central vacuole, we've got chloroplasts. Those are the big things that differ between the two. Now, when a plant cell is established and matures, and it has its cell wall built around it, it is like a brick in a wall. It is stuck in place. It is encapsulated by its cell wall. So in order to exchange things throughout an organism, throughout the entire plant, from cell to cell, you need holes in the cell wall so that you can access supplies or transfer things between cells. And these are called plasmodesmata. They're basically continuations of the cytoplasm between cells. All right? So they're highly regulated pores between adjacent plant cells, but plasmodesmatal regulation and all of that stuff is for a plant phys course, if you decide to take it, or for Botany 105, they probably talk about it a little bit. Any questions about the generalized plant cell? I love it. The central vacuole, we talked about it also. It develops, in the video they talk about it as a storage area, but it also helps the cell develop internal turgor pressure. Don't worry, I'm going to close that door, but I want to get a good slide up here. Oh, thank you. I appreciate that. If I had my clicker, I'd be able to walk over there, but no clicker today. All right, cool. Plasma membrane, phospholipid bilayer, hydrophobic region. I never know how good of a job I've done so far on this, but don't worry, our next presentation is entirely about the cell membrane, okay? Once we get through this cell overview stuff. So the cell membrane is a phospholipid bilayer with a bunch of stuff embedded in it. All right? These proteins that are embedded in the phospholipid bilayer, to a large extent, they're going to determine what can cross the phospholipid bilayer. All right? Remember, the phospholipid bilayer is a semi-permeable membrane. Only certain things can cross it. And what determines the things that can cross the phospholipid bilayer are like very small or hydrophobic molecules, because in the phospholipid bilayer, there's this region that's all hydrophobic fatty acid chains, okay? And like dissolves like. So look, there are cholesterols embedded in the phospholipid bilayer. A cholesterol is a special type of steroid hormone. Steroid hormones are lipids. Lipids are hydrocarbon chains. They're just in ring structures. So they're nonpolar. So they're hydrophobic. So they can be embedded in the phospholipid bilayer. This is the whole reason that we had to have any of our chemistry discussion, really, was so that you could understand what's hydrophobic, what's hydrophilic, what will get embedded in phospholipid bilayers, what can cross phospholipid bilayers. It's so incredibly important because the phospholipid bilayer is the primary barrier between the inside of the cell and the outside of the cell. That generates order. That's what you need to be alive, is some difference between the inside and the outside. Okay, a question and then a question. Yes?
Is it possible that we could be studying or seeing like hydrophobic or hydrophilic things on like the next exam?
Yes. Yep, absolutely. So there will be some osmosis questions and some like protein transport questions on the next exam. The study guide is also already posted in that week's module. I won't be changing the subjects, but I change the questions, but I don't change the subjects because I like the subjects that I test on in this course. I think I've got it pretty, down. Yeah, so you can already start looking at that if you want. Awesome. Question.
So the cholesterol, it gets embedded and then stuck in the membrane, or can it go through? Like, are
Great.
there things that just get stuck in there, or and then certain things that go through, or are there things that can do both? Because the phospholipids are hydrophobic and hydrophilic, so how
Yeah.
does that work?
That's a great question. I'm giving you 4 John points. I really like that question.
I really needed that kind of
Yep. Don't forget they are worth nothing and they expire every day. But
They expire?
yeah, I'm sorry.
Use them tomorrow.
Sorry, they expire. So check this out, right? Remember, all this stuff all of the fluid is an aqueous solution. So the outside of the cell, the extracellular fluid, and the cytoplasm inside of the cell, it's water-based. So cholesterol, being hydrophobic, isn't going to very readily dissolve into this extracellular fluid. So once it's embedded in the phospholipid bilayer, it's usually pretty stuck there. Because if it tries to squeeze out, it gets repelled back into the phospholipid bilayer, right? It can stay in the middle, but into the cell. Yep, exactly. Because everything is an aqueous solution.
But most things that transport in and out of the cell go through protein channels, correct?
If they are not small hydrophobic molecules,
Not small hydrophobic molecules in the protein channels.
yep.
Okay.
If it's small and hydrophobic, it'll just go bloop and float through here. And it will, it has enough energy that it will extend out, but most of the time it would be in the phospholipid bilayer. Of course. Now the one exception here, the very frustrating exception, is water. Because water is a polar molecule. It's very small. It's also everywhere. Okay? This is aqueous, this is aqueous. So water is going to move across the hydrophobic domain at pretty appreciable rates. But it's because while it has a resistance to moving across, it's everything. This is mostly water. This is mostly water. So water molecules do cross because they're small. Yeah. Question, please.
My question was, do they generally have to be small and hydrophobic, or just small or just hydrophobic?
Small encourages it, hydrophobic encourages it, right? So the combination of the two makes them move very quickly. If it's hydrophilic and small, it's not likely to cross. Something that won't cross at all are ions, because they're charged. They're so hydrophilic. It doesn't matter that it's a single hydrogen atom, a proton, right? Just a hydrogen proton. That's super tiny, but it's also incredibly hydrophilic. It'll just be surrounded by water molecules. So those things won't cross the phospholipid bilayer. So it's a combination of the two. Sort of makes sense? Awesome. And don't worry, I've got slides all about that coming up. Yeah.
So how does the cholesterol get into the bilayer then? It's held on both ends of the cell membrane.
So the cholesterol molecules are built up inside of other members of the endomembrane system, and then they get embedded into membrane, and then they get shipped, and then they fuse with the membrane, and now the cholesterol's there. Kind of makes sense? It's super weird, and of course, can we observe all of this happening? Not really, kind of, but only in snapshots. We can't see it happening in real time. So a lot of these answers are theoretical answers, you know? But I'm still going to present them as if they are fact because that's what we do, you know? Very good. And then later we'll just say, oh, we used to say this. Okay, awesome. I'm excited that there already seems to be some engagement in the importance of the cell membrane. Don't worry, we are going to talk about it ad nauseum in the next PowerPoint. It's what it's all about, okay? The nucleus is held by a nuclear envelope. We're going to talk about genetic structures as well in future PowerPoints, so we can sort of move past this one pretty smoothly, all right? In a eukaryotic cell, DNA is packaged into discrete units called chromosomes. In a prokaryotic cell, there's one chromosome. In a eukaryote, there's multiple chromosomes. All right. 23andMe takes its name from the 23 distinct chromosomes within a human. We've got 2 copies of each. We're diploid. All of that stuff will come back as we talk about genetics and DNA structure. This PowerPoint is meant to talk about the components of the endomembrane system, really. You know, that's what we're really trying to talk about. So the nucleus, it's enclosed by 2 phospholipid bilayers. It's got an outer and an inner membrane, all right? And it is connected to the endoplasmic reticulum. Here's an image of the nuclear envelope with the outer membrane partially removed, so you can see the outer and the inner membrane. You can also see these nuclear pores. These nuclear pores are important because you got to get things out of and into the nucleus. But they're also going to be tightly regulated because the nucleus holds all your precious, precious DNA. Question?
I just wanted to ask another question. Like, when you guys do like all this research and get these images, are they always in gray or like?
Great question. So I've done electron microscopy like 1 time, and it's its own field, okay? But they all actually have no natural color, because with electron microscopy, you never use light. You look at electrons that you blast through a sample, and you measure their scattering pattern. And then a computer interprets that scattering pattern and produces an image like this. And so if there was never any light involved, there's no possibility for color, right? So that's why they're always black and white, because you never captured light off this. You put this sample into a chamber that excluded light. And then when you see things like this, they're colorized. So this image was not collected with any color on it. Color was added to help tell the things apart. Satisfying?
Oh, that sounds like a lot of like imagination going on.
Yeah, you know, you have to trust the machine to be able to interpret the electron scattering patterns. All right, for example, yeah. But electron microscopy is its own field, and it's pretty advanced. And there's lots of reasons to, trust these images as accurate because they make good predictions and they look like the images that you can capture with light microscopy just zoomed in. You know what I mean? Yeah. If you want to get into electron microscopy, it's a career path, and we have classes here where you can do electron microscopy. I think there's 3 on campus right now, which is wild for a school of this size, thanks to some good work done by a lot of faculty who've written a lot of grants, All right, so the nuclear envelope. You had a question about this earlier. Wait, what?
I was just raising my hand, but my hand down.
Oh, yeah. No, I was just identifying that you had a question earlier about this, but you have a question right now about something else. How can I answer it? What's the question? No problem. I'm deducting all those John points. Yeah.
Your question, John Points?
I'm deducting them. Yeah, sorry. You should have had your hand up higher,
Sorry.
It's okay.
So
All is forgiven.
is in prokaryotic cells, we do not have this nuclear envelope because that would be a membrane-bound organism.
You're-
Organelle, right?
John points restored, exactly.
Thank you.
You're so welcome.
You don't understand.
I get it. The nuclear envelope, right, is a eukaryotic feature.
Because it's a membrane-bound organelle, so it has a membrane inside and outside.
It's membrane bound. It's a membrane bound structure because it's just a membrane bound structure, right? It's got a specialized function, but it's not necessarily an organelle. So don't panic too much about that, though. Just know that one of the main differences, where the names come from, for prokaryote versus eukaryote, is the nuclear envelope. The nucleus is just a membranous structure that encloses the genetic material. All right?
An envelope is like more structural support around the nucleus.
It's just called the nuclear envelope, and it's a double membrane.
Okay.
Yeah, the nuclear envelope is the name given to it.
That's only in eukaryotes.
Only in eukaryotes. Yep. Eukaryo means true kernel. The kernel is the nucleus. Cool? I love it. Awesome. Good, everybody? Okay. So check this out. Nuclear envelope is attached to the endoplasmic reticulum, so you can make the RNA product and then ship it directly to a ribosome. It helps to keep things directed. What are the ribosomes? The ribosomes are the protein factories, okay? In a ribosome, RNA is read, and the sequence of RNA is used to direct protein synthesis. So basically what happens is you read the DNA nucleotides, and you assemble a polymer of RNA nucleotides, and then the ribosome reads the RNA nucleotides and uses those to assemble a polymer of amino acids. And that's how you get a protein. So the ribosome, which we will talk about a lot more in the future, is an organelle, but it is not membrane bound. It's an organelle in that it is a combination of multiple types of macromolecules that carries out a specific function. It's like an organ in your body, which is a combination of different cell types and tissue types that carries out a specific function. It's just small. It's an organelle, all right? It's made of ribosomal RNA, so it's a special type of RNA, and protein. Okay? Some ribosomes are embedded in the endoplasmic reticulum. Some ribosomes are free-floating in the cytoplasm. The ones which are bound are called bound. The ones which are free are called free. All right? So these are bound ribosomes on the endoplasmic reticulum, and they give the endoplasmic reticulum a rough appearance because they've got all these, they're studded with all these ribosomes. So there's the rough ER, which is the part of the endoplasmic reticulum studded with ribosomes. And then there's the smooth ER. And I'll show you the textbook, definitions of like, what do they do? What's the difference between the rough and the smooth and what their function is, okay? But this slide is all about the ribosomes, okay, which are the protein factories. They read RNA and they assemble a polypeptide. How do we feel about that? Awesome. Thumbs up. I love it. I love it. Okay, we'll continue moving forward. This is all going to come back later. These pieces will all come up later. I put it on the slide to stop me from getting off on a tangent on it if we didn't have the time for it. So listen, we're going to talk about these things. Don't worry. Endoplasmic reticulum. Okay? So, the endomembrane system, what is it? It's all these membrane-bound things, okay? Nuclear envelope, ER, Golgi, lysosomes, vacuoles, plasma membranes. They're all connected to one another via transfer of vesicles, okay? Because it's all phospholipid bilayer. And remember, if you bubble a piece off of one phospholipid bilayer, it can fuse to another phospholipid bilayer and become part of that phospholipid bilayer. All right? You can just keep bubbling it off, moving it over here, attach it to a motor protein, walk it where it needs to go, all right, and then dump proteins or whatever's inside where they are supposed to be. Yeah?
So my question is, that seems like a pretty comprehensive list. What isn't in the endomembrane system?
The ribosomes, because they're not membrane enclosed, they're not really connected by vesicles. Peroxisomes, I think, are technically part of the endomembrane system. I just don't talk about them with you because they're like annoying. They're really, really annoying. Try looking them up. Try being like, what's a peroxisome? And you're going to be like, there's no information here. It's so infuriating. There's lots of stuff inside of the cell that's not in the endomembrane system, like proteins that are floating around. Cytoskeleton isn't a part of the endomembrane system. The cytoskeleton provides structure and transport tracts and all sorts of things. So the endomembrane system is everything bound by membranes. And it illustrates for you how important the compartmentalization is. I'm going to go 1, 2, 3. Sound good? How did I do with the answer to your question? Satisfying? I love it. 1.
So is this eukaryotic and prokaryotic?
Just eukaryotic. Just eukaryotic. Remember, huge delineating factor. The prokaryotes don't have all these membrane-bound organelles. So they've got no nuclear envelope. They have none of these. Except for lysosomes. And they have ribosomes, but those aren't membrane-bound. So they're not part of the endomembrane system. Isn't it great? So much to juggle all at once. Yes? Mitochondria are not listed as part of the endomembrane system because they don't like bleb off and feel. You'll see chloroplasts aren't in here either because they don't do the same thing of blebbing off little little vesicles and transferring material. They don't exchange. Here's the thing. They don't exchange membrane with the other members of the endomembrane system, except for when they're being assembled. Because the membrane had to come from somewhere, right? And it's usually like endoplasmic reticulum stuff. Yeah.
I know I'm asking a lot of questions, but
That's good. That's a good thing.
the vesicles. So are they like these bubbles that carry these things, but are they made of phospholipid bilayer?
Yes.
So they are like a membrane that carries, because those things would not be able to float freely due to the clarity?
That's a great way to think about it, right? The exterior of these things is hydrophilic. So they can float around in the cytoplasm easily enough, right? They can move through.
They transport it more efficiently?
Yeah, it lets it be more directed. It's like putting, I gotta get 80 toothpaste tubes, lids to the back of this room. And I could like just start throwing them back to the back of the room, or I could put them in a box and I could put them on a little track that just goes and like drives them back there using a little bit of energy. Way more efficient, way more directed. And that's the thing about the eukaryotic cells in general, is they have all this additional compartmentalization, which is energy intensive, right, to maintain all of this. And like it can lead to all sorts of problems. But you get higher complexity that's achievable. Yeah. All right. Endoplasmic reticulum. It's like most of the membrane in eukaryotic cells. It's a bunch of folded up membrane, all right? There are 2 regions. The rough has ribosomes studded into it. The smooth has no ribosomes studded into it, all right? Bless you. The endoplasmic reticulum, boom, okay, has it has different functions depending on if it has ribosome, if it's rough or smooth. All right. The rough has these bound ribosomes and it makes proteins. It's also really great because if you've got a ribosome stuck in the endoplasmic reticulum already, okay, you can make the protein inside of the endoplasmic reticulum and then just package it up in the ER and then ship that vesicle. As soon as the membrane pops off of the ER, now it's a vesicle. It was ER, it popped off, vesicle, carrying a protein synthesized by a ribosome embedded in the ER. Okay? There's also just a membrane factory. So it just makes more phospholipids. Okay? Makes more sheets of phospholipid bilayer, and when you're ready to assemble more organelles, of course, there's going to be processes regulated for that. Smooth ER. Yeah, it doesn't do anything as exciting, but it's all incredibly important. Especially, surprisingly enough, like the calcium ion storage is an incredibly important aspect of the endoplasmic reticulum's function. But you don't need to really worry about it. You'll take a cell bio class and you'll talk more about this. So now listen, you've got the endoplasmic reticulum, you've made a protein, you put it in a vesicle. It needs some more modifications. You ship it to the Golgi apparatus. The Golgi apparatus receives those proteins from the endoplasmic reticulum, further modifies the proteins. It makes some stuff on its own. It sorts everything up. It packages it into transport vesicles, which have specific labels on their exterior that's going to help direct where they need to go. All right, so this is the sort of shipping center. It gets products from the endoplasmic reticulum. We're neglecting the fact that there are free-floating ribosomes that are doing this in the cytoplasm as well, but that's just because there's so much to talk about, okay? So just in general, it receives things from the endoplasmic reticulum, it modifies them, it repackages them, and it ships them out to where they need to go. Some of these things are destined to leave the cell. Some of these things are destined to fuse with other organelles. Some of these are lysosomes that'll fuse with a food vacuole. You know, all sorts of things can happen here. Okay, I talked to you about lysosomes already, and I don't think we're going to do it again. All right? So we talked about phagocytosis, talked about lysosomes. Autophagy is when you fuse a lysosome with a vesicle or vacuole that contains some stuff that no longer works the way it's supposed to. If you've got a malfunctioning mitochondrion, you package that up in a vesicle, you fuse a lysosome to it, you dissolve it, you get the macromolecules out. Autophagy, self-eating. Cool? Phage relates to eating. Question, please.
How exactly does a cell organelle wear out?
So all of these things are constantly wearing down, right? They get lower stability, they accrue differences in pH, like in their interiors, right, that aren't fully controlled for. The membrane becomes less stable over time. The proteins embedded in the membrane start to lose their structure. You think about a mitochondrion, and what it really does is it creates a chamber of high acidity and a chamber of basicity, okay? And you would imagine that that's going to put a lot of strain on protein structures. So proteins are slowly degrading. Like with most things, like with something as simple as a leaf, as it ages, it becomes less efficient. But that is unlike me, because as I age, I become more deadly and dangerous.
Like wine.
Like wine, yeah. I'm like a deadly, dangerous wine. All right? Thank you. So there's, of course, more detail, and I kind of tried to avoid too much detail by deflecting with a fun gag. But yeah, there's so much to understand and study about how these things break down. But that's one way, is the proteins embedded in their membranes start to denature. Yeah. I love it. Okay. What do I got left with you? Let's say I got like 90 seconds left. That's long enough to talk about endosymbiosis. Don't worry. I've got a whole slide about this when we talk about cellular respiration and photosynthesis. I'm going to talk to you about endosymbiosis. But imagine this scenario, okay? You've got a free-living prokaryote. And that free-living prokaryote over time, and there's evidence of this occurring currently, there are organisms that are partway towards evolving a nucleus, where they're infolding their plasma membrane and using it to surround their genetic material. That protects it, all right? So now you've got this prokaryote that has a nucleus, and it's got its DNA all protected. It tries to eat a cyanobacterium, okay? Another bacteria, another prokaryote that can carry out photosynthesis. Maybe it has a problem with the enzymes in the lysosome or something like that, but it can't kill it, it can't dissolve it, it can't digest it. However, instead of that thing harming the host cell, the DNA is all protected, so it just lives inside the cytoplasm and sugars seep out of it. Well, now you've got a symbiotic relationship, and this is where chloroplasts come from. Same thing with mitochondria. You try to eat something that's really good at breaking down sugars, and you're not that good at breaking down sugars. But what you are good at is being a big old growth chamber that protects your own DNA. And so this is endosymbiotic theory, and the endosymbionts are cyanobacteria and aerobic bacteria in this case. All right? It's really cool stuff. We'll talk about it in more detail. That's the end of our lecture for today. I know we're like a week behind, but don't worry. We'll get through this on Friday. The exams are set up with this in mind. Goodbye, goodbye. Have a great day. Farewell.
Thank you.
Thank you.